So in this part we will be taking microbes in industrial products. And under this we will talk about beverages and antibiotics. So for growing the microbes for industrial products, the large vessels are used which are called fermenters to carry out the process of fermentation. Fermentations are large vessels where microbes are grown. So let us see how beverages are prepared using microbes. Beverages include wine, beer, whiskey, rum, etc. So for this beverages, for the preparation of beverages, the yeast, that is Saccharomyces cerevisiae, is used, and we have seen in our previous section about the yeast that it is used in baking purposes also. So when we make use of yeast for beverages, it is called as brewer's yeast. So let us see how yeast is used to prepare beverages like wine, beer, whiskey, rum, etc. So for this we need to recall the anaerobic respiration by yeast. For this purpose, the cereals like barley, wheat are used for the fermentation process where yeast grows and does anaerobic respiration, also called fermentation, which produces ethanol carbon dioxide and energy. The energy in the form of ATP. So this ethanol is the alcohol which is of two types. So when ethanol is prepared, ethanol can be converted into whiskey, rum, brandy by the process of distillation. Whereas ethanol without distillation is used as beer, wine. Now let us understand what is the difference between distillation and without distillation. Whenever yeast grows in the cereals, what happens? After reaching 13% of alcohol formation, yeast get killed up. Now you know the level of alcohol is in demand when a person take a beverage. So in distillation process, the ethanol level increases so that the effect of alcohol is observed and the drinks like whiskey, rum and brandy are prepared. Whereas in beer and wine, the percentage of alcohol is less. And here the percentage of alcohol is more. So let us recall what we have done. We have talked about that microbes in industrial products can be used in the process of beverages making and also in antibiotic making. And in beverages, the ethanol is prepared by the process of fermentation by the yeast called Saccharomyces cerevisiae, and that time it is called brewer's yeast. So when we make use of yeast in making of beverages, it is brewer's yeast, and when we make use of the yeast in baking industry, it is called as baker's yeast. So there is no difference between both the yeast, but the purposes are different. Now let us talk about how microbes are used in making of antibiotic. The term antibiotic is made up of two parts. Anti means against and biotics means living organism. So when a particular microbe is releasing a chemical which is used to kill a pathogen, then that chemical is known as antibiotics. So let us see what is antibiotic. Antibiotics are the chemicals which are produced by microbes and are used to kill the pathogenic microbes. To kill the pathogenic microbes. So pathogenic refers to those disease causing microbes. And we have variety of microbes which are pathogenic in nature. So let us talk about the discovery of first antibiotic, that is the NCERT question also. The first antibiotic was penicillin. Now, sometimes in NCERT, in board exam, it is asked about the history of penicillin. So, Alexander Fleming while working in his lab, with the bacterium Staphylococci, found out that something was stopping the growth of Staphylococci. We will understand this history with the help of diagram. So Alexander Fleming was working in his lab. He was growing Staphylococci on his culture plate, and he left the plate in his sink in his laboratory and was expecting that this Staphylococci will grow enormously throughout this culture plate. But it did not happen. When he came next day, he found out that the growth of Staphylococci was restricted. So he was believing that something was growing around this staphylococci which was stopping the growth of this bacteria. Then later on it was found out that it was a fungus called Penicillium. Penicillium notatum. So Penicillium notatum was secreting a chemical called antibiotic, which was later on known as penicillin, which is the first antibiotic. The full potential of this chemical was discovered by Ernest Flore, Chain, and during the World War II, it saved the life of many American soldiers. In 1945, three scientists, that is Ernest Flore, that is Flore, Chain and Fleming, won Nobel Prize for the discovery of penicillin and knowing its full potential. Penicillin and knowing its full potential. Now in our next video we will be taking up how microbes are useful in making of chemicals, enzymes and bioactive molecules.